Welcome students, I am Dr. Vanita Chandel, Assistant Professor, Amity Institute of Virology and Immunology, Amity University, Noida. In this module, you will learn about macromolecular blotting or more commonly known as hybridization, its types and its applications. This technique is used for the detection of pathogens from plants, humans and animals. Beside this, it also helps to determine molecular weight of the proteins, and in this module you will learn methods of blotting and what are the variations and similarities in three types of blotting. There are three types of blotting: Northern, Southern and Western. I would like to mention here that hybridization and microarray are based on similar principle. Difference is that in hybridization, probe is added after binding of DNA or RNA to the membrane, whereas in the microarray, probes are added to the glass slide first and samples are added later. Blotting or hybridization is of three types. Types of hybridization, again it is of three types: Northern, Western and Southern. So, let us discuss first the principle of hybridization. It is based on the principle that two strands of DNA get separated on high temperature and re-anneal when temperature is lowered. There are two important features of this process. The reactions are specific, that is, the probes will only bind to targets with the complementary sequence. Second, the probe can find one molecule of target in a mixture of millions of related but non-complementary molecules. The method is named after its inventor, the British biologist Edwin Sutherland. Other blotting methods, that is Western blot and Northern blot, that employ similar principles but using RNA or protein, have later been named in reference to Edwin Sutherland's name only. Southern blot is a method used in molecular biology for detection of a specific DNA sequence in DNA samples. Northern blot. The Northern blot is a technique used in a molecular biology research to study gene expression by the detection of RNA in a sample. With Northern blotting, it is possible to determine the particular gene expression rates during differentiation and morphogenesis as well as abnormal or diseased conditions. Northern blotting involves the use of electrophoresis to separate RNA samples by size and detection with hybridization probe, complementary to the part or the entire target sequence. The term Northern blot actually refers specifically to the capillary transfer of RNA from the electrophoresis gel to the blotting membrane. The Western blot, sometimes called the protein immunoblot, is a widely used analytical technique which is used to detect the specific proteins in a sample of tissue homogenate or extract. It uses gel electrophoresis to separate native proteins by 3D structure or denatured proteins by the length of the polypeptide. The proteins are then transferred to a membrane, which is typically nitrocellulose or PVDF, where they are stained with antibodies specific to the target protein. So, what are the different blotting membranes which we can use in the process? The solid support onto which the separated proteins are transferred is usually of two types, both of which bind proteins with very high affinity. Nitrocellulose membrane has excellent protein binding and retention capabilities, is brittle and thus it is usually less effective when blots need to be reused. Whereas PVDF membrane, it demonstrates superior mechanical strength, making it suitable for stripping or reprobing. Now, probe has a very important role to play in the hybridization. In a molecular biology, a hybridization probe is a fragment of DNA or RNA of a variable length, usually 100 to 1000 bases long, which can be radioactively labeled, and it can be then used in DNA or RNA samples to detect the presence of nucleotide sequence that are complementary to the sequence in the probe. So, labeling of DNA or RNA probe can be done by different methods. Most important methods are first radioactive labeling and second is your enzymatic labeling. Now, what are the procedures for Southern and Northern blotting? Procedure for Northern and Southern blotting is same except that in Southern we use DNA and in Northern we use RNA. Steps which are followed are: first genomic DNA is prepared or RNA is prepared, then they are restricted with endonucleases, denatured DNA with alkali, They are run on the agarose gel. From gel, DNA is blotted or transferred to nitrocellulose membrane and fixation is done in the presence of UV light or heat. Probe labeling is done either using radioactivity or enzymatic labeling. Then hybridization in various buffers followed by signal detection using X-ray film or antibody is done. There are terms like Slot-Blot or Dot-Blot hybridization. Now, Slot-Blot or Dot-Blot assembly is convenient way of transfer of DNA or RNA or just directly applying the plant sap to the membrane. These are the apparatus which are being used in the Dot-Blot and Slot-Blot, and images showing how it looks on the membrane after DNA is being transferred. To perform Dot-Blot and Slot-Blot transfer is the easiest way of transferring DNA RNA. We simply crush plant material, example if plant sample is a virus infected, and load that sample to the Slot-Blot or Dot-Blot assembly. Then after transferring sample to the membrane, pre-hybridization and hybridization procedure is followed. Probes are prepared from cloned viral cDNAs are used in the southern blots to detect the viral DNA in the infected plants. A modification in this technique is called tissue printing. It employs blotting of leaf squashes rather than the viral DNA to the nylon membrane directly. Now, use of radioactive material for preparing the probe has been the major limitation of this technique. So, to avoid this hazardous material, non-radioactive labeling techniques have been developed. Procedure of hybridization is same, but it vary in sample preparation. If we are using DNA, then we need to restrict DNA using endonucleases, and then we will run on the gel, and from gel we will transfer to the nitrocellulose membrane. If we are using RNA, there is no need to restrict. If we are using plant extract in Slot-Blot or Dot-Blot, we will crush them in a buffer and then load them directly to the assembly, and from assembly they will be transferred to the membrane. We can also use capillary transfer to transfer DNA directly from gel to the membrane. Northern blot. This figure is a diagrammatic representation of the Northern blot. Here the RNA is extracted from the sample, it is being run on the gel and it gets separated on the basis of its size. From gel, RNA is transferred to the membrane, nitrocellulose or PVDV, and fixed in UV light or by heat. It is then hybridized with the labeled probe and finally visualized on the X-ray film. This figure is a diagrammatic representation of the southern blot. DNA is restricted using endonucleases and are being run on the agarose gel. Now, separated DNA fragments are blotted to the nitrocellulose membrane. It can be done by further two methods, classical and a very commonly used technique by capillary transfer. In this, the gel is placed over the stack of sponge and above gel nitrocellulose membrane is placed, and over that stack of paper towels is kept, or we can just replace stack by some heavy book also. All this is placed in a tray of alkali solution. Now, through capillary transfer, DNA from gel moves to the nitrocellulose membrane and bind with it. After completion of the transfer, blotting paper stack is removed. The membrane is marked so as to indicate orientation of the rinsed gel, and the gel is further rinsed in 2x SSC, which is saline sodium citrate, for a brief time. The buffer is removed by placing the membrane between the folds of dry 3 mm sheet, but the membrane is not completely dried. The membrane is further wrapped in a cling film and placed under UV light in a UV cross linker for about 2 minutes for cross linking of the DNA to the membrane. Now, we can keep this blot at 4 degrees Celsius until hybridization is being done. These are the pictures of hybridization bottles where whole hybridization procedure takes place, and hybridization oven and rocker. Before hybridization, there is a pre-hybridization procedure which is carried out at 1 hour at 42 degrees Celsius in the pre-hybridization buffer. After pre-hybridization step, the probe is added to the fresh pre-hybridization solution and incubated at 42 degrees Celsius for overnight, and this step is called hybridization. Till now, you have learned about blotting, types of blotting, that is southern, northern and western, principles of blotting and detailed procedure of blotting. The membranes used in the blotting probes, their preparation and labeling of the probes. You also now know about the Slot-Blot hybridization. After this, there are different washing steps with the progressive stringency. Now, stringency washes are performed after hybridization to remove unbound by salt concentration, surfactant concentration, temperature and duration of the wash. After washing, the blots are placed on the Whatman filter paper to remove excess of liquid and wrapped in a saran wrap immediately to prevent it from drying and for the purpose of autoradiography. Development of the blot. The blots are exposed to X-ray films by putting them between two intensifying screens for autoradiography in a cassette at -80 degrees Celsius. The films are developed after sufficient exposure depending on the intensity of the signal prior to autoradiography as determined by a portable radioactivity monitor. Now, these are the kind of gel pictures of southern blotting you will observe after the procedure is over. These are the result obtained after the southern blotting. Now, for enzymatic labeling, there are Biotin labeled kits available. Since radioactive labeling is not preferred due to the side effect of the radioactive handling and use is, so enzymatic labeling is also available, though sensitivity of the enzymatic labeling is less than that of radioactive labeling. The Biotin chromogenic detection kits are convenient tools for the chromogenic detection of biotinylated nucleic acids and the probes. Biotin labeled probe target hybrids are detected with alkaline phosphatase conjugated streptavidin. Streptavidin AP conjugates bind specifically and irreversibly to the Biotin labeled probes. These probes are then visualized using a chromogenic substrate for alkaline phosphatase which produces a blue-purple precipitate. Therefore, visualization does not require X-ray film or any other specific equipment in this case. Streptavidin. Streptavidin is a biotin binding tetrameric protein from Streptomycin avidinii with a molecular weight of around 60 kD Dalton. The streptavidin biotin bond is the strongest known non-covalent biological interaction. So, formation of such complex is a very rapid process and once formed the complex is unaffected by external factors. Streptavidin AP. Streptavidin AP conjugates bind specifically and irreversibly to the Biotin labeled probes. The probes then are visualized using a chromogenic substrate for alkaline phosphatase. Alkaline phosphatase cleaves the substrate and this produces a blue precipitate and therefore visualization does not require X-ray film or any other specific equipment. Now, Western blot. This is a diagrammatic representation of a Western blot protocol. We run a denatured protein on a page, that is polyacrylamide gel electrophoresis, which separate components on the basis of molecular weight. After that, we transfer the proteins from gel to the nitrocellulose or a nylon membrane by passage of electric field. Now, membrane is probed with antibody and is radio-labeled or enzyme linked with secondary antibody and substrate to visualize color on the membrane. Western blotting procedure. This is a flow chart of Western blot procedure stepwise. Sample preparation is followed by gel electrophoresis and transfer of protein from gel to nitrocellulose membrane, followed by ELISA steps of blocking, antibody probing and detection. It is a detailed procedure from how to prepare a sample to detection. For sample preparation, the cells are harvested, washed and lysed to release the target protein, and after this, these proteins are being run on SDS gel. After electrophoresis, these proteins are transferred from gel to nitrocellulose membrane in a wet or semi-dry condition. For both kind of transfer, the membrane is placed next to the gel. The two are sandwiched between absorbent material. The system is connected to a power supply and blotting is run for 1 hour. Next step is blocking. Blocking is very crucial step in the immunodetection phase of Western blotting because it prevents non-specific binding of antibody to the blotting membrane. The most commonly used blocking solutions contain 3 to 5% BSA or non-fat dried milk in a solution of PBS, that is phosphate buffered saline. The next step is antibody probing. The blot will be incubated in a dilute solution of antibody, usually for a few hours at a room temperature or overnight at 4 degrees Celsius. The secondary antibody serves not only to act as a carrier of the label, but is also a mechanism to amplify the emitted signals, as many secondary antibodies can theoretically bind simultaneously to the primary antibody. Detection with substrates. The most common antibodies label which is being used in Western blots is HRP, a small stable enzyme with high specificity and rapid turnover. The signal is detected when HRP is exposed to a substrate solution in the final step of the immunodetection procedure. Substrate solutions for Western blotting are chemical reagents that are acted upon by enzyme to yield a signal that can be easily measured. More routinely, HRP is used with ECL for detection. For ECL detection, the substrate is luminol, which is oxidized by HRP in the presence of hydrogen peroxide to produce light. The emitted light is detected by exposing the Western blot to X-ray film or by using a CCD camera for light capture. The emitted light forms a band on the film or on the screen of the imaging system, indicating where the HRP labeled antibody has bound to the target protein. Now, these are the gel pictures showing gel photographs after Western blotting procedure. Now, what are the different applications of blotting? Southern in the mapping of genomic clones, estimating gene numbers and pathogen detection. Northern gene expression profiling, it is very commonly used, and Western is used for the detection of the molecular weight of the proteins. So, students, after this module you will be able to define blotting, types of blotting, principle of blotting, procedure of blotting and various applications of blotting. Beside these 3 types of blotting that are being discussed in this module, there is Eastern blotting also, which refers to biochemical technique used to analyze protein post-translational modifications such as lipids, phosphomoieties and glycoconjugates. It is most often used to detect carbohydrate epitopes. I hope you have followed what I have taught you. For further clarification, you can refer to e-text of this module and you can judge yourself by taking part in the questionnaire provided with this module. The details of this module are available on the UGC website also. Happy learning. Thank you.